Patterson here. Today we're going to go over the process of photosynthesis. In the last video, I went over all the players. Those are all the pieces that are working in photosynthesis. I really believe you need to understand those parts before we can really start talking about how all these pieces work together. So if you haven't watched it, I'll put down in the bottom, go back, watch that, look at all these individual pieces, and now we're going to talk about how all these pieces work together. So let's get started. The formula for photosynthesis is going to be products that are going to be made, and I'm going to have reactants that are going to come into it. So I have to put in carbon dioxide, CO2, and water, and then I'm going to have a reaction happen. And my products is going to be glucose and oxygen gas. This is a waste product of plants. Photosynthesis has to deal with plants. So plants are going to make glucose and they're going to produce a waste of oxygen. For me to be able to get this, I need to take carbon dioxide and I need to take water. So when we look at photosynthesis, this is what I need you guys to take away. I have to make glucose. I need C's, I need H's, and I need O's. So I need carbons, hydrogens, and oxygens. I need C, H's, and O's to make glucose. So I have to find my C's from carbon dioxide. I have to find my H's from hydrogen, and I need to find O's from my water and my carbon dioxide. I'm going to have a lot of leftover O's that I will not need, so those will be a waste product. So when we're looking at photosynthesis, I need to make sugars. So I need to find C's, H's, and O's. I need to harvest those in order to be able to get them to put them all together. And that's kind of like the big picture of photosynthesis. So photosynthesis, I have the process of converting light energy into chemical energy, and I have two different processes that happen. I have what's called a light dependent. I need light to get this started. Plants cannot live in a dark room. They have to have light. So I have to have light come in to start it. The second part is the light independent. I do not need light for this part. I need light to build and pull pieces off for the Calvin cycle to be able to work. But this does not need light. It's dependent upon light pieces, but it's not dependent upon light itself. So again, I need to make sugar. I need to get C's, I need to get H's, I need to get O's, and I need to put all of these together. So this part here is helping me harvest H's, and then I'm getting some O's, and over here I'm harvesting my C's and some more O's. So this is all a harvesting and trying to get material to where I can build my sugar over here. So let's look at the light dependent cycle. This is the part where I need light to be able to run this part of the machine. So if you remember, we have photosystem. These are my pigments. These get all excited when light photons hit, and remember they have different wavelengths. And if you forgot about that, go down and in the description are the players, and I go more in depth with that. So these are going to take these photons of light, get these electrons super excited. These electrons are now going to jump up and they're going to come to this electron acceptor and they're going to slide down this slide. Do you remember what this slide was called? This slide is the ETC. And these electrons are going to slide through, but then it loses energy here. These photons are going to re-excite these pigments, and these electrons are going to get pushed back up. This electron acceptor is going to push them back down the slide out to the end. NADP plus is waiting. NADP plus is going to grab a hydrogen. And you remember electrons? How many electrons is NADP plus going to grab? 2 electrons. So this is now going to be grabbing hydrogen protons and 2 electrons. And this now turns into NADPH. It's NADPH because I have the hydrogen attached. Remember I said this was like a taxi. This is going to come in and pick these up. And then this is going to go to the Calvin cycle. Now, where are these electrons coming from? They're coming from the water molecule. So water comes in, it's going to rip off these electrons. Hydrogen protons, now remember, I don't have any electrons, they're just hydrogen protons. These hydrogen protons are now going to be in this thylakoid space. So remember those Oreo cookies I showed you? in the last video, this is that red space in the middle. So these are going to start to build up. I'm going to have one oxygen atom is going to get pulled off, and that one oxygen is going to bond with another oxygen to make oxygen gas, O2. This becomes the waste product. This is going to leave the plant as a waste. So now I have my electrons going through. As they go through, they're going to have a negative charge. I have hydrogen protons out here. These hydrogen protons are very positive, and they're going to be attracted to this negative charge of this ETC. It's going to start pumping in these hydrogen protons, because remember, they don't want to come in here. I have a ton of hydrogen protons. These hydrogen protons want to leave. They want to go from an area of high concentration to an area of low. When they're being pumped in, they don't want to, so it has to be forced. So this charge forces more hydrogen protons into this area. For photosynthesis to work, I need this thylakoid space full of hydrogen protons. I want a high concentration here. So then what happens is these hydrogens want to leave, and they want to leave quickly, and the only escape they can go through is what's called ATP synthase. So if you remember, this is going to be an opening. These hydrogens are just going to flood through this ATP synthase. This ATP synthase is going to start to spin from the force of these hydrogen protons coming through. ADP and one inorganic phosphate is going to come into ATP synthase. This spinning motion is going to be kind of like a washing machine. It's going to spin super fast, and it's going to slap one of these phosphates onto the ADP. When it does that, I turn from an energy poor molecule now to this energy rich molecule. This ATP is now going to be used in the Calvin cycle to be able to make energy work. Remember, I said these are like coins, and I have to put these coins into the machine to make them work. So now I have my ETC is going to produce a electromagnetic charge that's going to allow it to come through. NADPH is now grabbing H's, because remember, what do I need to make sugar? I need C's, H's, and O's. So NADPH is bringing in my H's for this Calvin cycle for me to make my sugar. This ATP synthase, this hydrogen concentration right here, hydrogen ion concentration, is helping me make ATP, and ATP is what I'm going to use here in the Calvin cycle. So now I'm bringing energy, and I'm bringing H's into the Calvin cycle, and this is where I'm going to make my sugars. So the light comes in. Where do the electrons that move through the ETC come from? They're going to come from the water. This is going to produce the electrons. This is going to produce hydrogens, and this is going to produce a little bit of oxygen that will be a waste product. So what's the purpose of the electrons going through the ETC? This is to make a electrochemical gradient in order to allow these hydrogens to come into this system. They do not want to come in here, so it's going to force them in here. So I have a ton of hydrogens here. So then that way, when they want to leave through this ATP synthase. It's going to be spontaneous, and they're just going to just start flowing through. The energy is going to be enough to put a phosphate on an ADP to make ATP. And then my hydrogens will get pumped back into this thylakoid space. So those will go, NADPH will take my hydrogens to the Calvin cycle. So now I have light-dependent reactions. This is the part we just talked about. I have my photosystems, I have my ETC, I'm pumping in my hydrogen protons, I'm taking my electrons off of water, I'm making oxygen gas as a waste product, and all of these are going to be pumped through this ATP synthase for me to make ATP. NADP plus is going to grab a hydrogen proton and 2 electrons, and it's going to take it to the Calvin cycle. So all of this needs light to happen. This is what we call the light independent cycle. I do not need light per se for this. I need the products of the light, but I do not need light for this process to work. So let's start going into this. So for this one, I need you guys to just bear with me. You might have to watch this part a few times, but I need 3 carbon dioxides to come in. They can only come in one at a time, but I'm going to show all 3 here. So one of these carbon dioxides is going to come into the Calvin cycle. I have that, remember that enzyme Rubisco? I said it had 5 carbon molecules and it splits apart. It doesn't like to be with that 6th carbon. So Rubisco is going to come in. I have my 5 carbon Rubisco. This carbon dioxide is going to bond with this Rubisco. It's going to be attracted to it. So now I have 6 carbons, 1, 2, 3, 4, 5, 6. And these 2 oxygens are on here, but we don't show them. It gets too busy. So we're just going to show just the Rubisco with this extra carbon on here. So just pretend those oxygens are still there. I'm going to bring in another carbon dioxide. Another Rubisco is going to come in. And now I'm going to have 6 carbons. Another carbon dioxide is going to come in. And I'm going to have 3 Rubiscos now with this extra carbon. And remember, it isn't like this. It becomes very unstable and it's going to break. And now what happens is it's going to go in and it gets called 3-PGA. So now I've gone from Rubisco with that 6th carbon added to it. Now it breaks, and this broken Rubisco is now called 3-PGA because I have 3 carbons for each of these molecules, and I have 6 of them. Now we call this carbon fixation. The carbon has been fixed onto the Rubisco. Now what's going to happen is NADPH is coming in. Remember, we made this in the light-dependent cycle. I have a hydrogen and I have 2 electrons on here. So what happens is these hydrogens are going to be tagged onto these 3 PGAs. So this one's going to get a hydrogen, some electrons. This one's going to get a hydrogen and electrons. So I need to put those on there. And when I do, I lose that hydrogen and those electrons, and I get turned back into NADP+. This is now going to go back to the light-dependent cycle, and I'm going to have hydrogens re-added to it. The taxi is going back to the light-dependent cycle to add stuff to it. But I need 6 NADPHs for this to happen because I have 6 of these 3 PGAs. This cannot happen on its own. So for me to have 6 NADPHs, this is not going to just automatically go on there. I need help. You remember, what are the coins that help make a machine work? We need ATP. So ATP is going to come in and it's going to donate, it's going to apply one of these phosphates to this NADPH, and it's going to cause it to attach to this molecule. And then it's going to release that phosphate. It's going to turn into NADP, and now, ADP. It's going to turn into ADP, and I'm going to go back to the light-dependent cycle and go through ATP synthase and be made back into ATP and come back. So these will go back and forth between these cycles to bring in H's and the energy needed to add these hydrogens to these carbons and these oxygens. So now this is this 3-PGA with these H's on it is now being reduced. And when something gets reduced, electrons are being added. So remember, there's electrons on here, and these electrons are being added to these molecules. So they're being what we call reduced. So when they are reduced, I have hydrogens, I have electrons on here. We now call this G3P. So 3-PGA is when I just have carbons and oxygens. After I add the H's and the electrons and have reduced it with electrons, I now am called a G3P. This G3P is now going to take just one of these G3Ps and it's going to hold it off to the side. This is now going to be used to make glucose. But look, I'm only half a glucose molecule. Glucose is C6H12O6, and I only have 3 carbons. So I need 3 more carbons, another one of these, to be able to make glucose. But I just can't pull one of these because remember, I have to make Rubisco again. I've broken down my Rubisco. I need this enzyme for the Calvin cycle to happen. So we do what's called regeneration. So I'm taking these 5 and I have to regenerate and remake this Rubisco. So these 5 pieces are now going to be added together. I'm going to rebuild them with ATP. The energy from ATP is going to make ADP, and these are going to get rearranged back into Rubisco. Remember, Rubisco has 5 carbons. So here I have 1, 2, 3, 4, 5. So those 5 can make this Rubisco. ATP is going to add them together. I'm going to have 1, 2, 3, 4, 5. These 5 are going to be made into this Rubisco. ATP is going to put them together. And then I'm going to have 1, 2, 3, 4, 5. These 5 are going to be made into this Rubisco. So I have to have Rubisco to accept these carbon dioxide molecules coming in. I have to remake them. Then the cycle is going to start all over again. So I'll have 3 Rubisco, and then 3 more carbon dioxides are going to come in. Remember, they can only come in one at a time. One's going to come in, attach. One's going to come in and attach. One's going to come in and attach. They're going to break apart, and then I'm going to have 6 3-PGAs. Then NADPH is going to come in, and it's going to start tagging hydrogens onto these PGAs. Again, it can't do it by itself. ATP's got to make that happen. Then when I have these electrons and these hydrogens on, these now turn into G3P. One of these will come over, and then one of these will be used to make glucose. So then I'll have 2. I'll have this one and this one. I have 5 G3Ps. These will get regenerated or remade back into Rubisco. ATP is going to rearrange these back to where I'm going to have 5 carbons and 2 phosphates. I'll have another phosphate that's going to come in and add to that. And then the cycle will just keep working. So for me to do this, I need 6 turns of this machine to be able to make one glucose molecule. So here I'm going to use this one. These 3 turns of this will make just 1/2 of a glucose molecule. 3 turns again will make another 1/2 of a glucose molecule. And then those 6 turns will make 1. So those will end up coming together, and I will make 1 glucose molecule. Okay? So this is kind of more complicated. Again, if you understand the players, this is kind of going to help you out with this. This is the Calvin cycle. So carbon dioxide comes in, Rubisco is needed. I've got to have Rubisco to attach the carbon dioxide. It's going to become unstable and split. Then I have to add my H's and my electrons, because remember, for me to have glucose, I need C's, H's, and O's. So I have my C's and O's. I'm adding my H's, and now I have these molecules, but I can't use all of them. I have to rebuild Rubisco. So one of these is going to leave, and then these 5 are going to be rebuilt into Rubisco, and then I start it over again, and I'm going to have another 1/2 of a glucose molecule made. The other 5 will go back to being made Rubisco, and then I will have 1 glucose molecule. So I need 6 turns of this machine to make 1 molecule. All right. It's a complicated process. I would recommend go back, watch your players, understand how all of these little pieces work individually, and then come back and watch this, and now it should make a little bit more sense. I would draw it out. I would pretend like you're trying to teach this to somebody else and go through that process of photosynthesis. It's very complicated, but once you get it, it makes sense. If you guys have any questions, let me know. We'll talk to you later.